Hello, welcome to the demola, and today's lesson we are covering genetic screening. It's the last one for topic 2. We're going to talk about the types, why we do it, the ethics surrounding it. No time to waste. Let's get straight into it. Okay, so we're going to start with what is genetic screening, right? And as the definition, it's the testing of individuals or groups to find individuals or with an increased risk of specific genetic disorders. Now, they typically will not ask you to define genetic screening, but you do need to have an idea of what it is. I think really the most important parts are going to be about that you're trying to find an increased risk for specific genetic disorders. Yeah. When we talk about its main uses, it's literally identify individuals that have an allele for a particular disorder, screening embryos prior to implantation. We'll get onto that in a second, and also testing fetuses before birth. Another thing we'll get onto in a second. So, there are different types of genetic screening, right? I've just gone with this one. I haven't included it in the next slide because it doesn't have enough space and you don't get asked as much in your A level. So there's going to be identification of carriers. Now, typically this will be like a couple that's common. They want to see like the chances of the child having a specific disease that runs through the family, per se, sickle cell. Yeah. What they're going to do is they're going to analyze the DNA of both parents. They're going to find the probability that the future children will have the disorder. Remember, this is going to be similar to drawing your pedigree charts, right? It's independent for each child. So the chances of one child having a disease is the same as the next child, and so on and so forth. Right? The benefit of this is that it allows the partners to make an informed decision. So if there's a high risk of the child having a particular disorder, at least now they know. They can now think maybe we're going to have to go adoption, maybe we're going to have to go IVF. It just means that they have the right information to make a good. Okay, so now we're going to really get into the types of genetic screening. So these are the ones you typically get examined on. First one is PGD, pre-implantation genetic diagnosis. Yeah, involves IVF. So would have been covered in GCSE, I believe, but let's quickly run through it. IVF, in vitro fertilization. Essentially, what they'll do is they'll get a sample of eggs from the mother, and then they're going to fertilize them inside of a petri dish, yeah, using sperm from a donor or maybe from the father. Once those eggs are fertilized, they're going to see, first of all, which ones are fertile, which ones will survive in the uterine environment. And they're also going to now, if we're doing PGD, test the DNA, all right? They test the DNA, see which babies have the disease, which one don't, or which embryos have the disease, which ones don't, and then they implant the ones that don't. Yeah. What that means is embryos without the disease are implanted. The ones with the disease aren't. So we're avoiding potential abortions due to that reason. Yeah. The other one is prenatal screening. Very, very important that you are able to distinguish between chorionic villus sampling, CVS, and amniocentesis. So chorionic villus sampling, normally they're going to go through the urethra, and they're going to get to the placenta. They're going to scrape off some cells. Yeah. When they get the cells, they analyze the DNA of those cells, and then that will tell you, do they have this disorder, do they not? Yeah. Typically, we're going to carry this out 11 to 14 weeks. It does vary based on your textbook. I say anywhere between 10 to 14 is safe. Cool. And there's a 1 to 2% risk of pregnancy because you are getting quite close to the fetus. So a 1 to 2% chance of causing a miscarriage through chorionic villus sampling. Next, we have amniocentesis. So amniocentesis, what we need is, we need this fluid to form that surrounds your fetus. That's called your amniotic fluid. Yeah. You can go through the abdomen, you can go through the urethra, but either way, you get that sample of amniotic fluid, and there's cells in the amniotic fluid. We test the DNA of those cells. Yeah, it tells us the same information as CVS, although some diseases can be picked up on one and not the other. Yeah, we carry this out later because we need time for the amniotic fluid to develop. So 15 to 20 is safe. Again, you might see it different in the textbook. Yeah, this one has a lower chance of a miscarriage, only 1% this time. And that's because we are not getting as close to that egg. So there's less chance of us causing damage. Another thing we need to mention is the ethics of it all. So identification of carriers is the first one we talked about. Just know, literally for this one and actually for all of them, it's not always 100% accurate. Yeah, it's not always that they've screened for a disease, they found it there, it means the child has the disease. Sometimes they see signs of a disease, child doesn't have the disease. Sometimes they don't see signs of the disease, the child has the disease. Yeah, cool. They only give risk. It's not definitive. Yeah, not definitive. It's only going to give the risk. There's a BRCA1, BRCA2 gene, right, which is the risk of breast cancer. Just because someone has that gene or has the 40 allele does not mean that they're going to develop breast cancer. But it does definitely cause them a lot of stress thinking, oh shoot, I'm at a big risk for this. Yeah. And also just know employers might discriminate against people. They shouldn't by law, but they might. They might think if someone has this to a specific disease, maybe that means that I should be a bit more cautious about hiring them. That's the kind of mindset that we want to avoid. Now with PGD, the main thing that we need to say is designer babies. Yeah, this is the main drawback of PGD. Imagine a mother comes in with the father, with the partner, and they decide that they want a child to have blue eyes, they want a child to have brown hair. This is something we want to avoid. And if we select embryos, we can kind of almost lean into that. We do not want to do that. That's the whole concept of designer babies. And again, forced results. Someone could have bought a completely healthy baby, or that completely healthy baby is not going to have a life because their test results was wrong. And then pretty similar with prenatal testing, except for the fact that this one carries a risk of causing a miscarriage, as we covered in the last slide. And then ethical issues around abortion as well. So some would say it's murder. Some will say it's against religion. Some will say that you are taking away the right of the fetus to consent. All of those points can be valid in one way or the other. Just know them as ethical points to raise in a question for now. Okay, so it is question time. Now, first one, you're drawing a genetic cross for me, please. Similar to what we've done in other lessons. That's fine. And then now this is going to be more about this genetic screening, yeah? Name the prenatal test and why might the couple not have that test. So we'll leave both of the questions on screen for you to be able to see. Have a go, please, and we'll get into the answers. Okay, so we've answered the first one. Now, I needed to figure out, is Tay-Sachs autosomal dominant, autosomal recessive, is it sex-linked? What in this information tells me says the couple without Tay-Sachs disease, but the first child died from the disease, right? That must mean that both of them were carriers, yeah? So I'm going to write out the mother, father, I'm going to write out the genotype, then I'm going to write the phenotype, yeah? And then I'll draw my pedigree chart. Now, didn't write on. In this corner here, these are the only people that are going to suffer from disease, yeah? So if it's a quarter, and we're also going to say that TT is affected, the answer is 25%. Now going to the max scheme, CVS, it said 11 weeks, right? What we say, 11 to 14 weeks, mainly are going with CVS. And then also, when we talk about why they might not want to, they may have done research and known it's not always going to be accurate. Yeah, they may have done research and seen, or they would have probably been told there's an increased risk of miscarriage. False positives, termination of a healthy fetus. Yeah, and then also some people it will be against their values of belief. So make sure that you are happy with those points and you are able to kind of explain why someone might not want to go for some genetic screening. Cool, and now it's time for the A-star tips. So what do we want to do? We want to understand the different types of genetic screening and their indications. So we need to know when to do PGD, when to do amniocentesis, when to do CVS. Yeah. Make balanced arguments in the ethics questions. I think it's a bit hard when you do have a strong belief about something to have to kind of tone it down. But in the exam, you don't want to come across as too forward or too argumentative about your point. You need to give a balanced argument to gain more marks. Yeah. Link to benefits and uses of screening. I think it's very, very important, even outside of just trying to get grades on a test. Very important that you do promote things like this. And also just make sure you know the difference between PGD, amniocentesis, and CVS. Because that's going to lead us on to what not to do. A lot of people confuse these tests. Make sure it's not you. Personal viewpoints and ethical arguments. Again, pretty much what we've talked about. But yeah, you just need to make sure you're not actually saying what your point of view is. Even if the question needs you to answer that, use the data in the question. Yeah, don't use your own knowledge, use the data in the question. And also just remember, screening is not a diagnosis. Yeah, so For example, someone gets screened for breast cancer, doesn't mean they have breast cancer, but they might be at an increased risk for it. So I really hope that video was helpful for all of you. If it was, please like and subscribe. We hit our goal of 500 subscribers by the end of the year, which I'm over the moon about. Thank you so much to everybody who has subscribed and dropped all these really, really lovely comments as well. Any questions, comment down below. I'll do my best to answer. We've got the TikTok and the Instagram both at A Star Demola as well. And I'll catch you in the next one. Bye for now.